This protocol can be used to prepare fungal and plant materials for solid-state nuclear magnetic resonance, or NMR, and dynamic nuclear polarization, or DNP experiments for complex biosystem characterization. This technique allows us to investigate biomaterials to an atomic resolution level in their native environment, for example, in whole cells.
The acquisition of high-resolution structural information about fungal materials will aid in development of antifungal drugs. The method provides insight into composition and structure of complex carbohydrates in fungal cell walls and can be applied to many carbohydrate-rich organisms, including plants, fungi, algae, and bacteria.
Some investigators may struggle with fungal contamination in their cellular cultures. My advice is to sterilize the medium and equipment thoroughly before use to prevent this contamination. This demonstration will help investigators with little experience in NMR or cell culture to learn how to implement these techniques in fungal and plant systems.
To prepare unlabeled liquid growth medium, dissolve 6.5 grams of yeast extract peptone dextrose, or YPD powder, in 100 milliliters of distilled water, and then autoclave the resulting solution for 25 minutes at 134 degrees Celsius. To prepare unlabeled solid growth medium, add 6.5 grams of YPD agar powder to 100 milliliters of distilled water, and autoclave the medium for 25 minutes at